Hey guys, this is Kendall Terry, and this is part 2 of Mendelian genetics. So let's just jump right in here. So the last thing we looked at, I can back this up one, is the key terms that we use in genetics. We've been doing some practice with those in class, so hopefully you're more familiar with those and you feel comfortable with those as we go from here. So when we look now, Mendel, as he developed these key terms, as he developed and really analyzed what was going on here, he came up with 2 laws that we still use today in genetics. The first is called the law of segregation. The law of segregation says that for each parent there is 2 alleles. So for purple you've got big P, that was the true breeding parent. The F1 generation was big P, little p, and then the other true breeding parent we know was little p, little p. And whenever those 2 true breeders came together, we know that this one got that little p, big P came from this parent here. But whenever that F1 generation went to make the F2 generation themselves, these chromosomes separated from each other and went in opposite directions. So you end up with a gamete that has a big P and a gamete that has a little p. Okay? So the law of segregation is that those will separate from each other, and it's kind of like that coin flip idea. If I'm going to flip a coin and you get to call it in the air, whenever I flip that coin, you have to make a choice. You're either going to say heads or tails, and it's a 50% chance that you're going to be right. Well, the same thing here with genetics. Whenever we're doing genetics problems and you're dealing with an individual that has 2 alleles, one of those alleles will fertilize and develop for the offspring. The other allele may not, depending on the organism that you're discussing. It may only produce 1. If you're talking about humans, on average, that's going to produce 1. So only 1 sperm is going to unite with 1 egg. So each parent is going to donate 1 allele. It's a 50% chance of which one of those you're going to get. Then we look at the next law, which is called the law of independent assortment. In the law of independent assortment, what we're doing here is we're looking at different traits. Now, Mendel only looked at 1 trait at a time. And as he looked at 1 trait at a time, he went all the way to the F2 generation, added up those results, and he always got a 3-to-1 ratio. As he started looking at more than 1 trait, he noticed that on average, those traits assort independently of each other. So let's talk about what that means. So let's assume here that we've got the seed color. Okay, so seed color is yellow, big Y, and let's say little y. That's the F1 individual. So that would have been a yellow seed plant. And it's also a purple plant. So let's see if we can do that on here as well. Let me change it here. Okay, purple, big P. We're still in the F1 generation. So as those develop into eggs, okay, what you have to realize is that these 2 alleles, the one for seed colors and the genes or traits for flower color, are more than likely on separate chromosomes. And you got to think back to meiosis. But when those chromosomes line up in the cell, they are not attached to anybody else. They're only concerned with their homologous pair, which will have these traits right here on it. They don't care about the chromosome that has the seed color. So those will assort independently of each other. And that's called the law of independent assortment. We're going to do some practice with this in class to help clear up some of that. So to help us figure out what we're doing, that is a Punnett square. And we've already done some of this in class, but it's good to go over now. So let's look at the easiest one to look at would be the F1 generation. So in the F1 generation of flower color, we had big P, little p, crossed with a big P, little p. And then we put those into a Punnett square. So here is going to be the first parent. And we're going to look at the gametes. So the one possible gamete would be a big P. The other possible gamete would be a little p. This individual's gametes will go here. So we got big P and little p. I'm trying to make the difference there. It's hard. So then you simply take those traits and fill in your boxes. So if this gamete combines with this gamete, you end up with a gamete that is big P, big P. If this gamete combines with this gamete, you end up with a gamete that's big P, little p. If this little p gamete combines with this big P gamete, which we always do the dominant gamete first, it's big P, little p. And then we've got two little p's. Now this is actually what Mendel found in his F2 generation. And we know this to be the case because when you look at his ratio of a 3-to-1 ratio, he said that in his F2 generation he got 3 purple to 1 white. When you add up the phenotype of these boxes, this one, because the dominant allele is purple. This one, because the dominant allele is purple. This one, because the dominant allele is purple. This one, no dominant allele, recessive. So it's a 3-to-1 ratio. That is the phenotypic ratio. If you look at a genotypic ratio, you end up with 1 big P to a 2 big P little p to a 1 little p. So it all depends on which ratio you're looking at. I know in class we've primarily focused on the phenotypic ratio. The next will be some practice problems, and I want you to take a little bit of time to do these practice problems as we go through them. So the first is, what is the probability of getting blue eyes if the parents are heterozygous? So let's look at that. What's the probability of getting blue eyes if the parents are heterozygous? So heterozygous is the first thing we've got to understand. That is big B, little b, and blue is recessive, so that individual parent will be brown eyes. The other parent, big B, little b, that parent is also brown eyes. And we want to know the probability of getting blue eyes, so we simply make our Punnett square. Big B, little b, big B, little b. Oh, man. Okay. So we got big B, we got big B, little b, big B, little b, little b, little b. So now when we look at these, we've got brown eyes, brown eyes, brown eyes, blue eyes. We go back and look, it says, what's the probability of getting blue eyes? Well, the probability of getting blue eyes is a 1 to 4, or we could also say 25% chance of us getting a blue-eyed individual. Next question, what's the probability of getting big B, little b if parents are heterozygous for both traits? So you can go back to the cross that we just did and add that up, because the probability of big B, little b if the parents are both heterozygous, you can take time to do that one. And then the last one, what is the probability of getting little b if one parent is homozygous dominant and the other is homozygous recessive? So we've got a new condition there. So homozygous dominant, big B, and we're crossing that with a homozygous recessive, little b, little b. So when we do the Punnett square, what we're going to end up with is all of our individuals are going to be big B, little b. And you can do the Punnett square and see that I'm right there. So if we look at the probability of getting big B, little b, it would be 0. And in genetics, that's an okay answer to give. Some people shy away from that. It's okay to give the answer of 0. Just make sure that that is the correct answer. The last thing I want to look at really on this is the test cross. We'll pick up the dihybrid in just in the next video. So in the test cross, what we look at is if we have an individual that has, let's say, go back to flower color. So we look at an individual that is purple. Whoa, purple. And we say, well, what is the genotype of this individual? Well, the genotype could be really one of two things. It could be big. Oh. It could be big P Sorry, I'm having to correct the mistake there. It could be big P, or it could be big P, little p. So we're trying to know which one. So what we do is a test cross. A test cross is we're going to cross this individual that we don't know the genotype with a white flower in this case. White because we do know the genotype of a white flower. The genotype of a white flower is little p, little p. All right, that's simple. So now we can do this cross because if we have big P, if the original parent is big P, then when we do the Punnett square, all of our offspring, and you can do the Punnett square to justify this, are big P, little p. There's no white flowers. Although if the parent is heterozygous, what we find is that half of the individuals are purple, or big P, little p. Half of the individuals are little p, which would be white. So what we do there is in genetics, we'll do a cross of a dominant phenotype, purple, we don't know the genotype of it, with a homozygous recessive one, because we know the genotype of that, that's 2 little letters. And we look at the offspring then. If we get a large enough sample size and we have no recessive individuals, then we go, okay, that individual that we didn't know had to be homozygous dominant. But if we end up with these homozygous recessive individuals, then we say, okay, that individual had to be a heterozygote, because you cannot get a homozygous recessive individual unless that parent that you don't know the genotype of has that hidden recessive allele in there to donate. So take a minute, think about that. We'll talk about that a little bit more in class.